All right, if somebody would lower the lights, please. All right, questions? Questions from anything we did last time? Nervous system? No? Sure? Okay. I think we ended here, right? We're talking about xylocaine and letadine. The bottom line is that there are animals that produce toxins, right? And these toxins can either kill or stun a predator or prey. And tetrodotoxin, we talked about the pufferfish, novocaine and xylocaine are roughly based on the chemical structure of tetrodotoxin, only they're not as potent. Let's talk about refractory periods. A refractory period, so if I draw an action potential, remember time in milliseconds, and on the y-axis it's millivolts, right? It's the potential, negative 90, I don't know, negative 60, 0, plus 20, right? And let's say that the resting membrane potential here is let's say that this is the threshold. All right. If you take a look, and in your book I think there may be, if you take a look at, usually it's an area that's very much towards the end of the action potential. So you have this, and then you have, what would be this. Okay, so here's the action potential, here's the threshold. There's what's called an absolute and a relative refractory period. During an absolute refractory period, a super stimulus, meaning one that is larger than normal, meaning one that I could generate with a volt of a stimulator, okay? During this window here, right, we'll number this number 1, and we'll call this number 2. During number 1, a super stimulus will not cause another action potential to occur. During an absolute refractory period, no matter what you do electrically, you cannot cause an additional action potential to occur. All right? It has a protective effect on the neuron or the muscle cell. We're going to see this when we get to the cardiovascular system, especially with the heart, because the heart has a very large absolute refractory period. The muscle does. And it's very protective, because when we're talking about the heart, it's all about timing. There has to be a sequence of coordinated events that allow each of the chambers to pump and empty in sequence. And if you go out of that sequence, it becomes very, very likely that your heart is not going to do what it's supposed to do, right? So in that sense, the refractory period, the absolute refractory period in the heart muscle is going to be one of the biggest you'll ever see. Okay? And this could be for a muscle or a neuron here, this action potential, right? It can either be a neural or it can be muscle. In this second one here, so this would be the absolute. In the second spot here, you have what's called a relative refractory period. During the relative refractory period, which starts about here, during the relative refractory period, if I have a stimulator, okay, a machine that will generate current, and I can connect it to you or the cell, and I apply a super stimulus, not the same stimulus that generated this, that threshold, but something much stronger, I can actually get another action potential to occur. And not only can I get another one to occur, but the next one that occurs during this, in the beginning of this relative refractory period, will actually be stronger than the one that preceded it. Okay. So you do this naturally all the time. How do you do this? You do it via neurotransmitters, you do it via hormones, right? So if you think about it in the fight or flight response, which is a response that is designed, that has evolved to protect you, you can actually generate supra stimuli using neurotransmitters like epinephrine, or also called adrenaline, to cause this to occur. So you can generate the super current yourself. It's not an easy thing to do, but it's not that hard to do either. Or you could just hook yourself up to some kind of stimulator to do this. All right, so absolute refractory period, if I apply the current here, nothing happens. Okay, nothing happens. If I apply the current here, then I get another action potential fired sooner than it normally would, and it's going to be, its amplitude will be greater than that of the first one. You can play this game all day long, right? Do the same thing here. You get something like this. The next one's bigger. So when we get to muscle, we'll talk about this some more. How do you make a muscle stronger, right, without actually changing proteins or anything like that? What types of things can you do? The muscle has in it right now, all of you have this capability to generate more force, right? And one of the things you can do is you can apply an extra electrical stimulus during this refractory period, and if you keep applying them, then you will continue to generate more and more force until it plateaus. All right. So in one of the labs that you're doing in the muscle lab in Physio X, you're actually going to do this with an electrical stimulator if you already haven't done it. Okay? If you already haven't done it. And it'll show you this, and what will eventually happen is you'll get a plateau. So it only works so much, and then after that you can't do any more. So there's a limit to this system. But it's based on this relative and absolute refractory period. When we get to the heart has this massive absolute refractory period because you don't want this to happen very often. Okay, and when it does, there's only a really small window for it to occur. All right. So the absolute refractory period, what does it actually mean at a cellular level? This occurs during a period when the voltage-gated sodium channels are either already open or have proceeded to the inactivated state during the first action potential. Okay. Following the absolute refractory period is an interval during which a second action potential can be produced, but only if the stimulus strength is considerably greater than usual. So during the absolute refractory period, mechanistically, what is actually occurring is these voltage-gated sodium channels are already open, so you can't open them again. Or you're in the inactivation state, and if you remember what those channels look like, they have that ball and chain, and that ball and chain is slotted in there. You can't do anything when that ball and chain is there, and if they're already open, you can't open something that's already open. All right, that's what cellularly is going on. All right. So what does the refractory period do? It limits the number of action potentials you can generate. The heart is your heart cannot beat as fast as you can fire a neuron or contract a muscle. You can actually contract the skeletal muscle or fire a neuron many more times and much more quickly than you can the heart. Your heart has an upper limit. Generally, we say, as a general rule of thumb, if you want to know what your maximum heart rate is, right, you take 220 and you minus your age. So if you're 20 years old, in theory, okay, theoretically, your maximum heart rate is 200 beats per minute. You can fire a muscle, okay, and you can fire an action potential for a neuron many more times than 200 times per minute. That's not the upper limit. I have measured in a female, I have measured someone in the 222 range, and she was 20 years old. So it's a theoretical number. It doesn't apply to everyone. But that's pretty much the top end, right? You can't go, unless you're a rat or a really small animal. If you take the heart rate of those animals, their resting heart rate for a very small young rat or mouse can be in the 600 beats per minute. Right? They're very, very small. But that's not humanly possible. Okay? So there are limitations on this. All right, so you can see these refractory periods. Here they start the refractory period lower. So you can take my artwork for what it is. It's the general gist is there. Here the relative refractory period starts here. Okay, and you fire another action potential. And so you fire it kind of late, right? You fire it kind of late. And so this one ends up being smaller. And this is the beginning of the relative refractory period. If you were to fire it here, you've got a bigger one. So what ends up happening is you're right at the tail end of this relative refractory period, so you get another response. If you do it again at the tail end very quickly, you're going to get a bigger response, ultimately a bigger response. Same thing as what I'm showing you there. Just a little, this is slower. I'm showing you the extreme. This is on the slow end. Okay? And so you can see that this is a sub-threshold stimulus, right? Then you have the stimulus strength increases here. It goes down, goes down, goes down, goes down. So what is different about mine, the way I did it, and the way I described it, and this one, based on this? How are they different? Why did my very first action potential that occurred after my first stimulus, so we have the first one here, my second one, which would be this one, is actually bigger? And what does this tell you about it? What kind of stimulus occurred here compared to the one I applied? So we're looking at these two. This is the original one, okay? Here's my original one in green. Now, my second one is bigger. But this one, the second one here, is smaller. Why? The answer's right here. Why? What does that second part of the graph tell you? What's the difference between the two? Yeah. Yeah. So, well, what's the difference between? Why is this one actually shorter? If I had drawn this one, mine is bigger. It would come up to here. This one is little. It's in the relative refractory period. What's the reason for that? The answer is yeah. Yes. And you know that because this is the stimulus strength that caused this blue action potential. This is the stimulus strength that caused the next one, right? And so it took actually less to cause this to occur. And if you do it frequently enough, by the time you get here, this is roughly what? 1, 2, 3, 4. This is a quarter of the stimulus that this is, and yet this is bigger than this. The one that I applied, I went with the hammer. Okay? And mine was actually physically bigger than this. And so mine went off the charts right away. So this is what's showing you. You can use, if you do this frequently enough, right? Your system can become super efficient. You use less energy and you end up doing more work. So you don't have to necessarily apply the hammer like I did if you fire it more frequently. Okay? Yeah.
But can't you only get a second action potential or response from in the refractory period if your strength is greater? So what would be sense?
Well, if it's to get it to be bigger, it would have to be greater. But you can apply less of a stimulus during the end of that relative. So the other thing that's different here is you're applying this at the end, where I applied it here. Okay, so at the end it's almost full. So what happened here? Okay, that's a really good question. It's a really perceptive question. Right? Where are we at here? What is this line here? It's the resting membrane potential. Where are we at? So technically, what's happened to the cell here? It's what? It's not more polarized. It's hyperpolarized. When I did it, had we repolarized the cell yet? No. Right? And so you needed a super stimulus to do it. Here, the thing has already been repolarized. It's actually gone into negative. So if we apply the same stimulus here, this thing actually becomes bigger. If we apply this same stimulus here, you don't get a stimulus because this is too small. You see the difference? It depends how big it is and where you apply it within that period. The closer you are to fully repolarizing, the smaller the stimulus to get something to occur. The further you are from repolarization, the stronger the stimulus has to be to cause something to occur. So that's a very perceptive question. Very good. Excellent. Does that make sense to everybody? It depends. And a lot of things in physiology are, it depends. Okay? But ultimately, if you do it frequently enough, okay? Speed. So how do you apply this in a real way? If you're an athlete, right, the faster you fire, the faster you move, the more power you can generate. The stronger you get. This is what's happening, right? Just by applying the force more often, you can generate more power and strength. Speed. This is a speed thing here. So you're using less actual force, less actual energy to produce more force because you're just doing it faster. So this is the difference between someone who's powerful versus someone who's strong. Right? Someone who's powerful moves something, your body. Think of it in terms of like an Olympic level sprinter. They move, let's say, 200 pounds faster than the next guy or gal that moves the same 200 pounds. That makes you more powerful. Strength has to do with the amount that you move, right? The weight. So if I can move 200 pounds in 2 seconds and somebody else can move 400 pounds, right, in 2 seconds, they're technically stronger and more powerful. But if they can only move that 400 pounds in 4 seconds, they're still stronger, but they're not more powerful. Okay, so there's a time component to power that strength does not have. This is speed. This is what it's showing you. You're firing it more frequently, and just by doing that alone, you're going to make the muscle be able to produce more force, or you're going to make the neuron be able to produce more neurotransmitters. All right, so this goes through some action potential propagations. Some housekeeping stuff. In a skeletal muscle, the action potentials are initiated near the middle of the cell and they propagate out, right? Because a muscle cell, so what does that mean? A muscle cell is a cylinder. A muscle cell looks like a vein or an artery, except it's filled in, right? And an action potential for a muscle cell starts there and propagates out. For a neuron, it doesn't, right? For a neuron, the action potential gets generated at the axon hillock and goes away. This is a muscle cell. This is a neuron. This is at the middle. This is not. This is right at the end of the tip of the soma here in the initial segment. That's what that means. Another thing here, the larger the fiber diameter, the faster the action potential. The same thing applies to wires in the wall. Once you understand these concepts, right? Once you understand these concepts, they apply to all electrical activity. A larger wire is a better conductor because there's less resistance. So the larger the diameter of the nerve, the axons, the faster the conduction rate. Okay, so it's very, very thin wires have a very, very high resistance. Okay. But a very, very thick wire has a very low resistance. These are your wires, the axons, the nerves. So if you're generating a lot of speed in that axon, the axon must be big, thick, and it must be myelinated. The bigger it is, thicker it is, and the more myelination there is, the more speed you can generate out of that axon because the resistance is low. Myelin. Myelin is a fatty substance. Remember, it's made by oligodendrocytes and Schwann cells. Myelin is, at least in your brain, one of the raw materials you need are omega-3 fatty acids, specifically DHA, docosahexaenoic acid. And that's why they put it in baby formula. And it comes out of mother's milk, too. That's why you need it for brain development. That's why you need fat when you are very, very small, because the myelin can't be produced without the fat. You can't take a small child, unless they're allergic to cow's milk, and put them on skim milk for a year. And the reason that is because you need all the fat, because that's what they need to develop their central nervous system, right? Same thing here. That's the myelin. Right? You can't put a little kid on a low-fat diet. It's a bad idea. Don't do it. Right? You need the fat. Forget about the hormonal part. We're talking about the development of the nervous system. You need the fat to make the myelin. Without the fat, you don't make the myelin. All right. So, action potentials occur at the nodes of Ranvier. What the heck does that mean? Okay, so let's take a look at this real quick. Let's go back to this neuromuscular junction idea. We're going to keep coming back to this. So we have a motor neuron. This is the neuromuscular junction here. Remember, it's a special name for a synapse between a motor neuron and a muscle cell. Right? Let's say that this is a very big muscle, something you use all the time. It will have these sheets that cover parts of the axon. And these sheets are myelin. Now, these sheets are insulators. Right? These sheets are insulators. Just like there's insulation around the wires that come out of that socket. If there weren't, this building would probably burn down, right? It allows the electrons, in the case of the metal, to flow in one direction, because an insulator will prevent the electrons, it will prevent the ions, in this case, from leaving, right? So while it's in this insulation here, it can only move in this direction. Now, when it gets to the nodes, this is the nodes of Ranvier. When it gets to the nodes, technically it could leak out. Right? So at all of these nodes, it could leak out. So if you compared that to a motor neuron that's myelinated versus a motor neuron that's not myelinated, this thing is leaking out all the way along. And it's going in both directions, right? You see how much leakage there is here? The ions just flow. Why do the ions flow? What do we keep coming back to in this class over and over again? The path of least resistance. It goes down its electrochemical gradient. The path of least. So this motor neuron is going to conduct a signal much faster than this one, because this one has an insulative layer that prevents a lot of the leakage. The thicker the insulative layer, the less ions can leak. Yep.
What would happen if the entire axon was myelinated? Would any nodes?
Yeah, that's a good question. If the entire axon was myelinated, you wouldn't have any leakage. You wouldn't have any leakage. But the system, it's interesting the way it's at this core here, within the nodes of Ranvier, and we haven't gotten to this yet, but there are sodium-potassium channels. The density of sodium and potassium channels at this node versus everywhere else here is very, very high. And so we don't actually get much leakage there, almost no leakage. Because as soon as the signal comes down here and gets to this, where there would begin to be leakage, we open up 4, 5, 6, 10 times more channels than exist here, and that supercharges the signal. And so it moves so fast in some of these, right? 250 miles an hour, that it actually looks like the signal does this. It's called saltatory conduction. It jumps. It doesn't actually jump, but it moves so fast that it looks like it jumps. If it was completely insulated, you wouldn't. It would still go fast, but it wouldn't go this fast because you wouldn't have those extra channels that sort of like turbochargers. It's moving really, really fast, and if it even begins to slow down a little, the turbocharger kicks in and goes boom, boom. Anybody ever been in a car with a turbocharger or a supercharger? All they are is you step on the gas. Supercharger is part of the engine. Turbocharger is extra. It's added to the engine, right? What you step on the gas in one of these cars, and it's being like shot out of a rocket. Because what ends up happening when you step on the gas really, really hard, you bring in the turbocharger, the supercharger brings in a lot of extra oxygen, and what happens to the tachometer, that thing that goes from like 0 to 10, there's a red area and a black area, is it goes crazy, right? Because you bring in all of this extra oxygen, and oxygen makes the engine work more efficiently and more power. That's exactly what happens here. It's like you're stepping on the gas, and the turbocharger kicks in, and it basically accelerates the signal. Okay? So every time you get to one of these nodes, you get an acceleration. So does the speed of the signal from here to here, irrespective of how long this is, does it ever change? No. If this was a graded potential, then it would, right? But it's not. This is an action potential. Does the speed change from here to here? It does, but it's such the distances that we're dealing with in human beings are so short that technically you don't see a difference. You can't measure it. But technically it could, but it just doesn't. All right? So this is what's called saltatory conduction. What would happen down here is what's called your standard conduction. Right? There are sodium and potassium channels that open and close all the way along. The signal just goes down this way. The same thing happens here, except it appears so fast that it appears to jump. Normal conduction versus saltatory conduction. We're going to keep coming back to this. Okay? This is not something that we're just seeing once. We get to the skeletal muscle, we'll see it. We get to the heart, we're going to see it again. Okay. That's what it says here. Yep. Myelination, talked about it. So the myelination, another thing I did talk about, is it reduces the metabolic cost. So not only does it make the signals move faster, but it does it with less energy. So a non-myelinated axon is slower, but costs you more energy to do whatever the activity is. Let's put this in the real world. When you first start doing something, you're slower and less efficient at it. You actually use more energy when you first start doing that activity. As you get better and better, one of the changes that's occurring is more and more myelination is occurring. It's not the only change, it's one change. And as that occurs, you become more efficient at it and actually use less energy. Who do you think uses more energy on a 1 mile run? Someone who runs every day or someone who hasn't run in 20 years? Same 1 mile run, same speed. You do everything exactly the same, except that one's a runner and the other one's been sitting on the couch for 20 years. Well, the guy that's been sitting on the couch for 20 years is inefficient. He actually burns more calories per foot than the person who runs every day. Because the person who runs every day has a lot of these changes that have occurred that have made him or her more efficient. Same thing here, it's happening at the nervous system level. Exactly the same. So you have a gross level, the person level, and then you could, a lot of these changes, you can trace them back all the way to the cell, right? And you can see that the same thing is happening at a small level like this that's happening at the entire organism level. All right, so here you were looking at the action potential. This is a motor neuron, right? On the out, what's happening here is, remember that the axon here is an extension of the cell. And inside the cell is more negative than outside the cell. So what happens when an action potential is generated? That flips. And this is due to fast sodium channels, voltage-gated sodium channels, the ones that the tetrodotoxin block. And so you see this, you see how there's a flip-flop here? You went from being negative on the inside and positive to the outside, and that whole thing flips. As you move along the axon, right? As you move along the axon, this is repolarized, this is depolarized. And you see how there's a shift, there's a switch between negative and positive? There's a flip-flop. And that's because sodium is flowing in here at a very high rate, which is changing the membrane potential. That here, it's already repolarized. Here, it hasn't depolarized yet. So this looks just like this. And then you keep moving. So as it moves along, right, it depolarizes the sections. Depolarizes the sections. Does everybody understand that? Does that make sense to everybody? What's actually physically going on here? Depolarization, the channels get adjusted.
So those, this is obviously action potential, if it's a graded one, is that where it loses?
Yeah, so if this was graded, this can't, so this could never be graded, right? This could only be an active event. But let's say that this was an area that was around here, and this was just happened to be the length of a cell, okay? Then this would lose its intensity as we went further and further along. An action potential, by definition, doesn't lose anything. It actually does, but you can't measure it. Right? So there is a difference between these two, but it is so slight. If this was graded, it would be massive, the decrement. And so this can't be graded ever. You can't transmit a signal down an axon with a graded potential. It can only move via an action potential. Right? You can't. Can't be done. This is never graded. This is slower than this, but they're both action potentials. An action potential, remember, is all or none. A graded potential is a stimulus that's trying to get you to threshold. This is already at threshold. Once it's at threshold, it goes. So that's a concept that's a little tricky, right? That's a concept you have to struggle with. You got to read it, you got to think about it, and you have to make sure you really, really understand what I'm saying, because I get it. Some people get it, but not everybody actually understands what that means. You really need to understand the difference between graded and action potentials. If you don't, you need to figure it out. We need to talk about it, because we're going to keep coming back to this over and over again, and they're not the same. Very, very different. Yeah?
Maybe you said this before and I just mentioned that. So can a graded, or does, or could a graded potential turn into an action potential?
No.
Or are they just two totally
They're totally different things. But can a graded potential get you to threshold to create an action potential? Yes. That is correct. They're not the same thing. Once it's an action potential, it's no longer a graded potential. So you can't, so you don't turn it in, right? But you can generate an action potential from a graded potential. So the wording is a little tricky, but it does make a difference. It does make a difference. Good question. Excellent. Very good. Okay. So this looks at saltatory conduction, which is what I was drawing. So all along the axon are sodium and potassium channels. All along this axon are voltage-gated sodium channels. Okay? So you have, when the initial segment here starts this process, it opens up a few sodium channels that are voltage. And then that gets amplified. A few open up many more, open up many more. So once this thing starts rolling, you have lots and lots of sodium channels opening at one time. But the initial part, until like with a graded potential, you can open up a few here and there. But once this starts, there's a lot of these opening. One of the limitations of skeletal muscle contraction and nerve conduction is not just how fast you open the sodium channels, but how fast you can close that, right? You've got to be able to close them, open the potassium channels, and repolarize. So it's a combination of how fast you can depolarize a cell and repolarize a cell. Because if you can open up the depolarizing ion channels, sodium, very, very quickly, but you can't make the potassium channels open and close any faster to repolarize the cell, then you're limited. Right there, it's a limitation of how fast you can get this thing to occur. So you have to be fast at both ends, the on and the off. On and the off, on and the off, on and the off. Okay? So this is showing you what it would look like with a jump, right? This is a node, this is a node. It appears that the signal jumps, but it doesn't actually jump. It's actually moving along this the whole time. And at this place here, what they're not showing you is these sodium channels and potassium channels are here also. They're just at a lower density. So if in this span you have 4 sodium channels, in this span you may only have one. So in this entire span, which is double this, you have 2 sodium channels, and in this span you have 4, right? So the density is 4 times greater. They're just not showing them to you, but they're there. So it doesn't actually jump. It seemed like it. If you took anatomy with me, we did Dora, jump, Salta, right? It doesn't actually jump, but it appears to jump. Any questions on it? So there's a disease that occurs where there are a number of diseases where you cause demyelination, multiple sclerosis, right? Which is a disease that you can get. It happens primarily in men, but not always, about my age, in the 40s, and is a demyelinating disease where the myelin sheath starts to break down and scar over. So either you can't conduct a signal or there's less room to conduct. So if we scar over this, right, and let's say this amount of tissue is scarred over, what have we essentially done to the diameter of this part of the axon? You made it smaller. What did I tell you about a smaller diameter wire or axon? Does it have a higher resistance to conduction or a lower resistance? A higher resistance. That's going to slow the speed down. And that's what happens. And you take drugs for it, and it makes you herky-jerky. Like, they'll be fine for months at a time, a year, and then all of a sudden they'll have this scarring occur, and all of a sudden they can't control. Like, they'll go to pick this up, and normally they can just pick it up and put it down. Then they'll have this occur, this scarring, and all of a sudden they'll go, and they can't get to what they want because they can't control the nervous system anymore because the signal speed is changing. And you have to adapt to that speed, right? You're used to a very high rate. And when it slows down, your brain and your hand are calibrated for a certain speed, right? Now you've got to recalibrate the system. And you're used to saying, I just can grab this. And now the speed has changed. You don't know it. So these two, this and your brain, they don't talk to each other the same way anymore. Because the speed has changed. Now you're like, all right, so then you super compensate and you, on this side, and you sort of do, and then you grab it and you're shaking and you don't have the same coordination. That's what happens in multiple sclerosis. Right? So it takes time, but it will kill you eventually, right? Because ultimately the scarring occurs to the diaphragm muscle axons, and the diaphragm muscle is a skeletal muscle, right? It's a dome-shaped muscle that's under your rib cage that causes you to breathe. And if you can't get the signals to go to the diaphragm and you can't breathe, well, then you can't stay alive. And that's ultimately what causes death for a lot of things, ultimately. But it's via the scarring mechanism. That's what multiple sclerosis is. All right, so this is that table I talked about the other day. You should look this over, know the difference between graded and action potentials, right? There's good stuff in here. Convergence, so synapses. This is the third section now of this chapter. Synapses can either be convergent or divergent. So in this case, you have 4 neurons that feed into one. Where would this occur in the body? Your cerebellum. Your cerebellum, the small brain that sits right back here under the cerebrum, but over the brainstem, and it's where a lot of the coordination occurs. Your cerebellum has tentacles. It's like the CIA. It sees everything. Okay? All of the information about everything that I am doing right now, everything I'm doing, every body part that's moving, is all converging on cells in my cerebellum. So there are neurons within the cerebellum that are seeing everything, and they're processing all of that. Okay? Remember that most of the neurons in your central nervous system are inner neurons. They're processing with brains. We could do something else. So, for instance, with skeletal muscle, right, with neuromuscular junction, you can have one motor neuron that diverges and controls lots of different muscle cells. This is actually called the motor unit. So you have different size motor units. We're going to talk about that when we get to muscle, right? So small motor units may have 10 of these that diverge. The large motor unit may have thousands of them. The bigger the motor unit, the more force that's produced with the activation of one motor neuron. So these are examples of where you can see each one. Is that all right? Okay. So synapses, again, this is sort of review. Remember, they could be a synapse is that point of connection. In this case, it would be here. There's no physical connection between a motor neuron and a muscle cell, a motor neuron and a gland, right? A neuron and a neuron. The synapse can occur between the axon of one motor neuron and the soma. It could occur axon to dendrite. It could occur dendrite to dendrite. So there are all kinds of exceptions to these rules. There's a nice picture in your book. Now, I have a different version, but there's a nice picture of this, roughly, give or take. In mine, it's figure 6-26. It's on page 160. It's probably on a different page, but it shows you the different kinds of synapses that occur. You should take a look at it because they're in all different places. Usually, it occurs between, the end of an axon and a muscle, the end of an axon and a dendrite. But it can be in lots of different ways, and so they show you the different shapes and the different positions where this occurs. There are 2 types of synapses. One is called electrical, the other one is called chemical. The ones that I have been showing you here are chemical synapses, meaning that it takes a chemical, a neurotransmitter, to bridge one end and the other. An electrical synapse is called a gap junction. Essentially what an electrical synapse is, I'll draw one for you, at least I'll try, is a small set of tubes. So if you have, for instance, 2 neurons that touch one another, all right, so this is nucleus of one, nucleus of another. They touch each other physically. You can connect them via a gap junction, and a gap junction is this small set of tubes, connexons and connexins, that physically allow the ions and the electrical current to flow through the tube. So it allows the electrical current to flow. You don't need a neurotransmitter to go between these two because there's a physical connection. Here, you need a neurotransmitter, remember, because the electrical signal can't jump this gap because this is insulated on this side. Can't do it. Here, there's no insulation. There's just a tube the ions flow from one end to the other. So this is an electrical synapse. It's called a gap junction. So this is called a gap junction. And this is a chemical synapse. So chemical, and remember this is called the neuromuscular junction. In this case, it has a specific name. Question? Oh, okay. Questions? Are you okay with that? Okay. This is pre and post. This is exactly what I just said here. So you can, again, just read a lot of this here for you. Read it afterwards. Okay, we're taking a look. This is again the chemical synapse. A lot of this, you should know this terminology, right? This is the terminal end of the synapse, the verticosity. This is the presynaptic side. This is the postsynaptic side. Here's the cleft. The whole thing is the synapse. All right. This is going to become important when we get to the neuromuscular junction. So I'm setting you up for muscle. A lot of what I'm doing now, when we get to muscle, will be revealed. When an action potential comes down this axon, okay, it's an electrical signal that comes down here with these sodium, voltage-gated sodium channels turning the signal on, and the potassium channels turning the signal off, all the way down. By the time it gets to this verticosity, what ends up happening is you have these calcium channels. All right? They're called N-type. N as in Nicholas. They're called N-type calcium channels. They're voltage activated. When that current comes down here, right, the voltage changes here. The voltage sensor says, oh, and it opens up these calcium channels. When calcium comes into the cell here, it fuses to this thing. See, these things are vesicles. In this case, this is the neuromuscular junction. These vesicles are filled with acetylcholine, the neurotransmitter. The calcium binds to synaptotagmin. Right? It's part of a snare protein. Right? It's part of this protein here. Synaptotagmin binds calcium. That calcium then actually activates these snare proteins, which is also connected to this vesicle, and the snare proteins dock. They physically connect the vesicle to the membrane. Without the calcium, this synaptotagmin doesn't turn the snare proteins on. They won't dock. They can't physically attach. You can think about these snare proteins like talons on a wall, right? The talons don't come out unless calcium's around. And if the talons don't come out, it can't hold on to whatever it is it's trying to hold on to. But as soon as the talons come out, they attach. And there's a fusion between this vesicle and the plasma membrane. Okay? So this is the very first time in the system, when we talk about the system is going to be, it's called excitation-contraction coupling, which we're going to get to in muscle. We've got an entire sheet on it. This is the first time you see calcium. Okay? The first time you see calcium, it's not the last time you will see calcium. If you don't have enough calcium, this process doesn't occur. If these don't fuse here, you can't get a muscle contraction. That's a problem. So anything that blocks this here causes problems. Botulinum toxin, botulism, will cause this problem of this fusing occurring in what are called excitatory synapses. So the motor neuron here has other neurons that attach to it that are excitatory and inhibitory. Okay? And botulism will cause these excitatory neurons here not to be able to transmit their signal. And so this won't reach threshold to chorotoxin. Clostridium tetani, tetanus, right? Everybody's had a tetanus shot. Does the same thing, except it affects the inhibitory neurons. So how are they different? If you get, anybody ever, I hope nobody here has ever used Botox. You're way too young for Botox. Okay? But Botox is botulinum toxin. Botulinum toxin is the most potent toxin on the planet. The smallest amount will kill you. So when you take Botox, you're taking your life in your hands because it's pretty darn potent. Okay? Botulinum toxin, what does Botox do to people's faces? So stars do it all the time, right? Famous people, people have lots of money. What does it do to the muscles in their face? It relaxes them. Imagine if what the right dose would do to your body. It would paralyze you. That's how it kills you. Tetanus, right? Clostridium tetani, similar mechanism, but what does tetanus do? What does it cause to happen? What physically happens if you get tetanus? Let's say you didn't have a tetanus shot. You stepped on a dirty nail. You could get tetanus from anything. You don't have to get it from a dirty nail, right? First thing, you go into an ER and you stepped on something sharp. First thing the doctor, the nurse, or the PA is going to ask you, when's the last time you had a tetanus shot? I don't know, right? So, we just give you another one. Why? Because if you get tetanus, if you see pictures of this, right, you will start having contractions that you cannot stop. And they will continue to happen. And so you see pictures of people, drawings from like the 17th century of tetanus, they look like they're disfigured. So it will continue, and what starts to happen is, first, you're paralyzed because you're contracting. You can't stop the muscle contraction. And then if it keeps going on, it keeps getting worse, then you'll start snapping vertebrae in your back. You'll start snapping your own bones. So before you die, you're going to start snapping all of the bones in your body, and then to boot, on top of that, then your diaphragm will stop because you can't contract it anymore. So then you won't be able to breathe anymore. Perfect. Just wonderful. So why do they ask you that? That's why they ask you that. Because you have to have one every 10 years. And if you haven't had one, you come in and they say, well, sir, madam, when was the last time? You're like, hey, I don't know, George. They just give it to you, right? Okay. All right, I think we will stop here. All right, so thinking forward, we're a week away from the exam. We have 2 more lectures. We are definitely not going to get through muscle. Okay, so the exam will be through neural. But Physio X, because you've had so long to do it, will be through muscle. But the lectures will only be through the nervous system. Okay, so that will be next Friday. 2 more lectures. Have a great weekend.